In gametogenesis, we will study both, that is spermatogenesis and oogenesis. So we'll start with first spermatogenesis. Now here we have already seen the diagram of the seminiferous tubule. So two types of cells were present in the seminiferous tubule. One was the germ layer, that is the germ cells, and the other was the Sertoli cell, which were required for the nourishment of the developing sperm. Now in spermatogenesis we are only concerned about the germ cell. So suppose this is the germinal epithelium or the germ cell. This is what? This is the germinal epithelium or the germ cell. Okay. Epithelium or the germ cell. Now the spermatogenesis is divided into three phases. First one is the multiplication phase, then the growth phase, and final is the maturation phase. So here we are talking about the multiplication phase. Now in multiplication phase, the type of cell division which is occurring is mitosis. So these cells will divide and produce several cells by mitosis, and this is what? This is the multiplication phase. This is what? This is the multiplication phase, in which cell division is occurring? Mitosis. Mitosis is the equational division, and the cells which are produced here are diploid. These were also diploid cells. So these are also diploid because mitosis is only occur, and these are called spermatogonia. Spermatogonia. Spermatogonia, you can say that they are the sperm mother cells, as we have seen that microspore mother cell and megaspore mother cell in the second chapter. So these are spermatogonia. Now these spermatogonia will be divided into two types. Suppose one is the X type and the other is the Y type. So the X type will be the stem cells, which will behave like the stem cells, which will multiply and produce more and more spermatogonia when required, and the type B cell will undergo the other phases of spermatogenesis. So suppose we are taking here this cell. Okay, now this cell will undergo the second phase, which is the growth phase. So in growth phase, it just enlarges itself. So this enlargement is due to the accumulation of the nutrients, but these nutrients are not from the Sertoli cells. These nutrients are the nutrients which are secreted by the germinal epithelium. Okay, these nutrients are the nutrients which are secreted by the germinal epithelium. Okay, so this is the growth phase. This is what? The growth phase. So growth phase is nothing but just the increase in size and the accumulation of the nutrients. Okay, now this cell which is formed here is called primary spermatocyte. This is called primary spermatocyte. Now this primary spermatocyte will undergo the next phase, that is the maturation phase, and that will take in account the division, that is meiosis, that is the reduction division. So this primary spermatocyte will undergo what? Will now undergo the first meiosis 1. Okay, and it will form two cells. It will form two cells. Now these cells will be haploid in nature. These cells will be haploid in nature, and these are called what? These are called secondary spermatocyte. These cells which are formed by the first meiosis are called the secondary spermatocyte. Now it will undergo the second meiotic division. Okay, it will undergo the second meiotic division, and so this cell will form two daughter cells, and these cells are nothing but, we call them as spermatid. These cells are spermatids. These cells are what? These cells are spermatid. So this phase, till here it was the growth phase, and from here to here was the multiplication phase. Multiplication phase. And this is here meiosis. Meiosis is occurring. That is a what? That is a reduction division. So we are getting what? We are getting what? We are getting 4 haploid spermatids. We are getting what? We are getting 4 haploid spermatids. Now there is a specific process in which these spermatids will now attain a particular structure, that is of sperm, which will have a head, a neck and tail. Okay, this is the sperm, and this process of formation of sperm from spermatid is called spermiogenesis. It is called what? It is called spermiogenesis. Okay, don't confuse it with spermatogenesis. Spermatogenesis is the production of spermatids, and after that when they acquire a particular typical structure of sperm, this process is called spermiogenesis. So the overall thing which we saw here is that in spermatogenesis the germinal epithelium will undergo the multiplication phase and it will form diploid spermatogonia. These spermatogonia will undergo the growth phase, the second phase, that is the growth phase, and that is the formation of the primary spermatocyte. It is also what? It is also a diploid structure. Okay, no division was occurring here, only the size increased due to the accumulation of the nutrients which were secreted by the germinal epithelium. After that, meiosis 1 will occur. Here it was meiosis 2. Meiosis 1 occurred and there was formation of 2 daughter cells, and these were called secondary spermatocyte, and the ploidy of the secondary spermatocyte was haploid. This is diploid and this is also diploid. So in examination they can ask as what is the ploidy of the secondary spermatocyte. Okay, so it will undergo meiosis 2 and 4 daughter cells will be formed, and these will be the spermatids, and spermatid will undergo a specific process, that is spermiogenesis, due to which they will form a structure, that is sperm. Okay, so the conversion of spermatid into sperm is called spermiogenesis, and the spermatogenesis process is divided into 3 phases, that is the multiplication phase, the growth phase, and this was the maturation phase. Maturation phase, that is the meiosis. Okay, so this is all about spermatogenesis. Now when sperms acquire a particular, when the spermatids, the circular looking cell which you saw, acquire a particular structure that is of sperm, what events are happening in conversion of the spermatids into the sperm. So see, these were the 4 spermatids which were produced, and these will be converted into, in spermiogenesis, will be converted into the sperm structure. So what were the events, what are the events which will help in the formation of these cells? So first one is the condensation of the nucleus. In this, the nucleus will lose its RNA and specific acidic proteins, only DNA and basic proteins will be remaining. Basic protein. So this is typically the genetic material only. The second is the Golgi. The major part of this Golgi will be present in the acrosome. And it will be helping in formation of the sperm lysins, that is the enzymes which will be responsible for the degradation of the outer layer of the ovum. Okay. The third one is the anterior portion. Anterior portion is pointed, flat. Okay. The fourth one we can say that the whole typical structure of the sperm is covered with the plasma membrane. Plasma membrane. The whole typical structure will be surrounded by plasma membrane. The cytoplasmic structure will only be present in the tail region. So when the sperm reaches the ovum, it will only give its genetic material to the ovum. No cytoplasmic content will be required in the fertilization process. So the cytoplasm is only present in the tail region, because there is no requirement of that cytoplasm inside the ovum. That ovum is already having its cytoplasm, which will provide nourishment to the developing zygote. So cytoplasm will only be present in the tail region. Sixth one is that the centrioles are arranged in the particular area, that is the neck region. The centrioles of this cell are arranged in the neck region. And the mitochondria which are present in a spiral or spring form. So these events will help in developing the structure of sperm, that is the conversion of the spermatid into the sperm, that is spermiogenesis. Okay, these were the spermatids and these are the sperms. Okay, so this was what all events will occur in the conversion of the circular looking cell into a particular structure, that is sperm. Now we'll see the hormonal control. Now we'll see the role of hormones or the hormonal control of spermatogenesis. Now you are aware of pituitary hormone, hypothalamic hormone. Okay, so at puberty, when the male reaches puberty, when the male will reach hormones. When the male will reach puberty, a specific secretion will occur, that is of gonadotrophins. Okay, specific will occur of gonadotropins. These are the hypothalamic hormones. Okay, this is released by now these gonadotrophin hormones will stimulate the secretion of pituitary hormones. Pituitary hormones, that is LH, luteinizing hormone, and follicle stimulating hormone. Okay, first at puberty the release of gonadotrophins will occur from the hypothalamus of the brain. Now from this these gonadotrophins will help in the stimulation and secretion of certain hormones of the pituitary gland. Okay, so the pituitary will now form LH and FSH. Now here this LH will act on the Leydig cells, or you also call them as interstitial cells. Okay, the Leydig cells will help in the secretion of androgens. Okay, this follicle stimulating hormone will act on the Sertoli cell, will act on what? On Sertoli cell, and it will help in release of certain factors which will help in the process of spermiogenesis. Secretion of certain factors which will help in spermiogenesis. Also the release of the sperm from the Sertoli cell or from the or from seminiferous tubule is called spermiation. Okay, so this is the role of hormones, that is when the male attains puberty there will be release of gonadotrophin hormones from the hypothalamus of the brain. Now this gonadotrophin will stimulate the secretion of pituitary hormones. These both are the pituitary hormones, LH, luteinizing hormone, and follicle stimulating hormone. The luteinizing hormone will act on the Leydig cell, and follicle stimulating hormone will act on the Sertoli cell. And these secretion of the Leydig cells, that is the androgens, androgens will be secreted by the Leydig cells, and Sertoli cell will help in secretion of certain factors which will help in process of spermiogenesis. So this is a very important topic, both spermatogenesis and oogenesis. This is the structure of sperm. The spermatid, which was formed in the spermatogenesis process, undergoes several changes in its structure, and it's now called as sperm. So what are the structures? You can see here that plasma membrane is given. Plasma membrane is given. Whole of the sperm is covered with the help of this plasma membrane. Now three structures you can see in the sperm, that is the head region, the neck, and the tail region. Okay, this is the tail. This sperm head which we are talking about contains an elongated haploid nucleus. Okay, nucleus containing chromosomal material, and it contains DNA and what I told you, basic proteins. The anterior portion of it is covered with a cap-like structure. See, you can see on the top of the sperm a cap-like structure. This is what? This is acrosome, and this acrosome is filled with enzymes which will help in the digestion of the outer layers of the ovum. The middle piece will possess numerous mitochondria. Okay, see, mitochondria are present here, as you can see, it will possess numerous mitochondria which will help in production of energy for the movement of tail. That will be essential for sperm motility. Okay, so this is the structure of a typical human sperm. The human male ejaculates about 200 to 300 million sperms during a coitus, of which for normal fertility, at least 60% of the sperm must have a normal shape and size, and at least 40% of the sperms must show vigorous motility. And this vigorous motility is provided with what? Provided with this mitochondria, which will serve as the powerhouse for this sperm. And Golgi apparatus here is converted into what? Is converted into what? I told you major two portions are there. One portion will provide with the production of those enzymes which will be present in the acrosome and which will help in the digestion of the outer layers of the ovum. So this is the structure of what? This is the structure of sperm. Oogenesis, the formation of ooted or egg. So oogenesis is generally a discontinuous process, and this we'll get to know why it is called as discontinuous process. And some very beautiful facts about oogenesis are there. Now, when the baby girl is in the womb of the mother, that is the first month of the gestation period, some 1700 primary follicles are formed. Now these primary follicles will multiply and proliferate, and by the second month of this gestation period, some 6 lakh primary follicles are formed. See, I am talking about the female baby, or we can say the fetus, the female fetus, which is present in the mother's womb. So you can see the beauty that the growth of the follicles that are responsible for the formation of the gamete is started when the fetus is inside the mother's womb. So when it's just first month, 1700 primary follicles are formed. Then in the second month, 6 lakh follicles are formed, and when the fetus reaches the 5th month, some 7 million primary follicles are formed. Now after that, these primary follicles undergo a specific process that is called atresia, that is the shrinking of the primary follicles, and till birth only some 2 million primary follicles are remaining at birth. Because of atresia, that is the shrinking of the primary follicles, only 2 million follicles are remaining. Now by the time the female infant reaches the puberty, that is the puberty, there is again the shrinking of this primary follicles, and finally some 60 to 80,000 primary follicles are remaining. Okay. When we talked about spermatogenesis, we saw that the spermatogenesis started at the puberty and it is an ongoing process. But in females, as there is a specific number of eggs in the lifespan of the female, there comes a phase where these primary follicles will be over, and this stage is nothing but the menopause. So in female this menopause is seen, and in male the spermatogenesis process is going in entire lifespan of male. This is the reason, as here we can see that till puberty only 60 to 80,000 primary follicles are remaining. So if a particular number of follicle is present and after years this particular follicle will get degraded, so a stage will come in female's life, that is the menopause, when the menstruation will cease. Okay, so this is the particular the amount of the primary follicles formed when the female is inside the mother's womb until the puberty of that female. Okay, now we'll come to the main process, that is oogenesis. As we saw in the spermatogenesis, there were 3 phases. First one was the multiplication phase, the other one was the growth phase, and the third one was the maturation phase. So the multiplication phase is nothing but the mitosis, that is the equational division. So the primary follicle which are remaining will undergo the multiplication phase. Okay, the primary follicle will undergo the multiplication phase, that is the mitotic division. Okay, so these are called what? The primary follicle is 2N and it is called oogonia. Oogonium. Okay, this is what called oogonia. That is for singular and plural. Okay, oogonia. Now this oogonia will undergo the multiplication phase and it will form primary spermatocyte. Primary. Here we are talking about oogenesis, so it will be the primary oocyte. Okay, it will undergo formation of many primary oocyte. Okay, now this formation of the oogonia is the multiplication phase. Okay, here this development of the oogonia into the primary oocyte is the growth phase. Now how this is growth phase, I will tell you. In this primary oocyte, when you will compare its size with the oogonia, it is very large. Okay, now the size of the primary oocyte as compared with the oogonia is very large. Okay, this size in humans, this is the growth phase, and this size increases some 200 times in humans. For every organism it's different, like in Drosophila it's 90,000 times, in frogs it's 2,000 times. Okay, so growth phase is the phase where the oogonia is converted into the primary oocyte, and this primary oocyte is large in size. This is because it will finally provide the nourishment, and according to the requirement of the nourishment, the size of this primary oocyte will depend. Okay, now this primary oocyte will undergo what? It will undergo the next phase, that is the maturation phase, and this is nothing but the meiosis. So first meiosis 1 will occur. Now we know that the sperm was not having any cytoplasmic content. Okay, so the egg should have a lot of cytoplasmic content so that after fertilization it can provide nourishment to the developing zygote. Okay, so also we want only one egg. There is no requirement of the other egg. So suppose this is the nuclear division, first it's occurring, and now the cytoplasmic division will occur. So the cytoplasm will be distributed unequally and there will be formation of two cells, and this is a small cell and this is a large cell. So this one is called the first polar body. Okay, this is called the first polar body and this is called the secondary oocyte. Okay, this is the first polar body and the secondary oocyte. Now this secondary oocyte will be in same stage till the sperm reaches this secondary oocyte. After, as we are learning oogenesis, so we assume that the sperm reaches the secondary oocyte and it will undergo the second meiotic division, meiosis 2, and it will form the second polar body, and this is the ooted or egg, or you can also call ovum. Okay, so there are some basic procedures or steps which we have to keep in mind. As this meiosis, when the particular cell is in meiosis, this will be arrested in the diplotin phase of prophase 1 of meiosis 1 till puberty. So all the follicles which were present, all the oocytes which were present will be arrested in the diplotin stage of prophase 1 till the organism, till that baby girl we are talking about attains the puberty. Okay, and this particular step, that is meiosis 2, will only occur when it is fertilized. Oh, you will not say fertilized, when this is encountered with the sperm. When this secondary oocyte is encountered with the sperm, then only the meiosis 2 will occur in this, and there will be the formation of the second polar body and the ooted, that is egg or ovum, in which the nucleus of the sperm will unite and there will be the formation of what? There will be the formation of the zygote. Okay, so these two events you have to keep in mind, that here the growth at birth will be arrested in the meiosis 1, that is diplotin phase of prophase 1 of meiosis 1 till the puberty, and after that when it attains the puberty, the further events will occur, like the formation of this secondary oocyte, and when the sperm is encountered with this secondary oocyte, the further division, that is the meiosis 2, will occur, that is the formation of second polar body and the ooted. So when we talk about ploidy, this will be the haploid and this will also be the haploid stage, and when the sperm encounters with it, it will become diploid. So this from here to here is the maturation phase, that is the meiosis reduction division occur. So we divided the oogenesis into 3 phases, that is multiplication phase, growth phase and maturation phase. Here growth phase is important because a large amount of nutrient, that is enrichment of the cytoplasm, is occurring, so that it can provide nourishment to the developing zygote afterwards. As we know that sperm was not having any cytoplasmic content, so all the nourishment which is to be provided to the zygote will be provided with the help of the the oocyte, all the granulosa or follicular cells which are present, which are nutrient, that only will provide, or all the cytoplasmic content which is present inside the cell will provide the nourishment to the developing zygote. Okay. So at maturation phase, first meiosis will occur, secondary oocyte and first polar body. Here nourishment is required, so one cell is very small, so we call that as first polar body, and there is no requirement of this cell. And again, when this secondary oocyte will encounter sperm, will encounter what? It will encounter that sperm, then it will form the second polar body, and the oocyte or the egg will be formed, and this will be fertilized with the nucleus of the sperm. So this is all about oogenesis. These two topics, that is spermatogenesis and oogenesis, are very important. So very important for examination. So you have to be thorough with these